So we'll start the class. So last week we were trying to study about meiosis. Okay, and just we do a quick recap of what we have done so far so that you'll understand it exactly. Okay, so we started with meiosis. We studied that meiosis has two parts. Meiosis 1 and Meiosis 2. Then we studied that Meiosis 1 has two parts. The karyokinesis 1 and then the cytokinesis 1. Then you have karyokinesis 2 and then cytokinesis 2. So in karyokinesis 1 you would have studied about prophase 1, metaphase 1, anaphase 1, telophase 1. Again here you have prophase 2, metaphase 2, anaphase 2 and telophase 2. So then we studied about the branching of this prophase 1. We started with this thing. We started to study about what is meiosis. Meiosis is a type of cell division that is seen in germ cells or reproductive cells in our body. So here in this meiosis 1, this prophase 1, we saw it is very elaborate. It consists of 5 phases. What are the 5 phases? Leptotene, zygotine, fascitine, diplotene, and diakinesis. So we studied about what happens in each of these phases in the last class. Okay, so we studied about like what happened in prophase 1. We tried to study. Okay, we have studied in detail what happens in prophase 1. So I was trying to tell you about your thing. I mean like how the crossing over happens. Okay, so let us just take quick some notes here. Okay, so prophase 1 is the longest phase in entire meiosis. It is of it comprises of 5 substages: leptotene, thin threads of chromosomes are formed. Zygotene, the homologous chromosomes pair up together. Facetin, the crossing over happens between what is the first dash? What will be the first dash? The crossing over happens between what and what? Dash chromatids. We have a term, yeah, non-sister chromatids. Very good. I'm seeing good number of you are answering. The crossing over happens between non-sister chromatids of what? What chromosomes? Homologous chromosomes. Very good. Good. So this is fascinating. Homologous. It is not homogeneous. It is homologous. It happens between non-sister chromatids of homologous chromosomes. Okay. You write one more point. The place of crossover is called chiasma. Okay, I'll just show first slide. Some people are asking. First, quickly write it. Then now I'm going to go for the second slide for some time. If you want to take down, you please take down. Now let me go to the third slide. So after facetin, what happens? You have diplotin, the chiasma starts to separate after the crossing over. And diakinesis, the chromosomes separate after the crossover. Thambi, all these things are happening inside the cell. Okay. In every cell, whenever a cell is about to divide through meiosis, the cell will undergo these changes. Okay, chiasma will be there inside the cell. So please understand that all these things are happening inside a cell. Okay, so you have this thing, everything will happen inside the nucleus. Okay, so everything will happen inside the nucleus. So when all this prophase is happening, this you also have to write something. While prophase 1 happens, 1 is complete. The nuclear membrane gets disintegrated. The cell organelles are lost. Okay, so this is what is happening when your prophase 1 happens. Okay, so after that, what would have happened? The prophase, by this time, the centriole would have happened, so it would started to give out the spindle fibers. The spindle fibers will start to come. Okay, and by the time, what would have happened? So you have had, let us say that this is a chromosome. Let us say this is father's chromosome. Father's chromosomes duplicate along with mother's chromosome. Okay, chiasma is formed. It is not like kind of a cell organelle. The place where these two chromosomes get joined is called as chiasma. Okay, when you're crossing over, no, you have, let us say that this is one chromosome, and there is this another chromosome which is crossing over. This place, this joint is called as chiasma. So here you will have like one more thing. Okay, so basically this joining area is called as chiasma. The place where the crossing over happens, the joining position is called as chiasma. Are we clear? Okay, tell me this thing, is my screen lagging? Okay, good. Many of them are not having, for some people you are having some issues. So I think it's just the normal network issue. Okay, good. So after this happens what? This is what happens in prophase. Prophase 1. Okay, when the cell started, when the prophase started, it would have been like this. When the prophase started, the cell would have been like this. But as the prophase 1 is getting completed, the cell would have become like what? This. Okay, so which means the prophase 1, the entire, all the 5 stages would have happened, and after the 5 stages, you would get a structure like this. Then comes your metaphase. So what happens in metaphase is the question. Okay, so now the arrangement will be such that, let us say, this is there. The spindle fibers will form like this. So what you are able to see is, you are not seeing like this. The arrangement is not like this. So let us say that this is one chromosome, and then this is another chromosome. These two are homologous chromosome. They are not arranged in this way, but rather it is arranged like this, such that one of this thing will go towards one side and the other thing will go towards other side. Okay, so while you're talking about a cell, if you're having 23 homologous chromosome, what will happen is, like, let us say that this is the cell. The homologous chromosome pair 1 will be like this, the pair 2 will be like this, pair 3 will be like this, pair 4 will look like this, pair 5 will look like this, pair 6 will look like this, pair 7 will look like this, pair 8 will look, pair 9 will look, pair 10 will be looking like this way. So what I'm trying to tell is like that all the 23 chromosomes, 23 pairs will be arranged in such a way that one half, like, it will be like such a way that one half will go towards one end and the other half will go towards another end. Okay, the arrangement is this way. Whereas in mitosis, how the arrangement will happen, the mitosis, there is no such thing. So all the 46 chromosome 1, 2, 3, 4, 5, 6, 7, 8, 9, up to 46 will be arranged in one line. Okay, so there it is arranged in one line. So there, how will it be arranged? The 46 chromosomes, the original will be going towards one side, duplicate will be going towards another side. That way mitosis will happen. Whereas in meiosis, the arrangement is of this pattern. Are we able to understand? This is meiosis. Whereas in case of mitosis, all the 46 will be arranged in one plate. 1, 2, 3, 4, 5, up to 46. This is mitosis. Okay, so now let us write metaphase 1. The spindle fibers connect with the chromosome. Ma Harita, you are like kind of typing something. See, there is a problem with the centromere of the chromosome. So please send your brother outside while your class is going on. The bivalents are arranged in a pattern such that they move towards opposite. It appears as if two equatorial plane is formed. Basically, it is arranged this way. This refers to one bivalent. This is a bivalent. This is another bivalent. These two are arranged this way. This is bivalent. This is bivalent. All these four things together, these two things together, we call it as a tetrad. Okay. If you're done, we can move on. Anaphase 1. The chromosomes get separated. The chromosomes do not get divided, not split in this stage. They just move towards the polar region. Okay, so listen here. What will happen here is okay, I have used a different color code here. So what is happening is they are moving towards the opposite poles. So spindle fibers will pull the bivalents towards the poles, or we can call them as dyads also. Okay, so this thing is called as a dyad. So this dyad is kind of pulling it out that side. So what is happening is this part is being pulled towards one side and this part is being pulled towards another side. The chromosomes get separated. Okay, what is happening? These two are getting separated. Or like you can say, instead of using the word chromosomes, you can use the word tetrad. The tetrad gets separated. Okay, but you can use the word tetrad. The tetrad gets separated. That would be a better way to put it. Okay, and the chromosomes are not split in this stage. What is the meaning of it? These two are not split. So It is like kind of like one dyad is moving towards one thing. They just move towards the polar region. The they here refers to the dyad chromosome. Are you able to understand? So basically, two dyads will make one tetrad. Okay, the dyads are separated. Or bivalence. We call, instead of using the word bivalent, I can use the word dyad also. And all these four things together, we call it as a tetrad. So whenever I'm saying that chromosomes are not split in this thing, what do we mean by that? Here, this division is not happening. It is just that one group is going towards one side. If you use the word, some of these words, you will get confused. So better is to use these correct words. Are we understanding? So the next one is telophase 1. The nuclear membrane reappears. The nucleolus reappears. The spindle fibers are lost. The chromosome gets uncoiled. So what exactly happens during this phase is, like, you are able to see that you are having two nucleus, and then in one side you are seeing there is MW. What are you asking, Niranjana? I'm not able to understand. The nuclear membrane reappears, the nucleolus reappears, the spindle fibers are lost, the chromosome gets uncoiled. A single cell has Uncoiled means getting unwrapped. So uncoiled na if it is like together as a chromosome, this is called as a coiled structure. So when it is like kind of like loosening up, it is like opening the thread, unwrapping. Okay, a single cell has like how many? Has two nucleus now. This is telophase 1. If you're done, you can just tell me so that I can move on. So after that, cytokinesis 1. The cytokinesis is division of cytoplasm. Two cells are formed at the end of meiosis 1. Okay. This is cytokinesis 1, so it is divided. So one cell was there, it underwent two nucleus, and then it has divided into two cells with two nucleus. So with this thing, what has happened? Meiosis 1 has completed. Okay, someone is asking for 8th slide, so please see. No, no, no. I did not pause. I'm just like kind of giving some space to write. If you're done, you let me know so that I can move on. Okay, so if you're done, we can move on. So there is a phase called as interkinesis. So here no, after this meiosis 1 is completely over, before the meiosis 2 starts, there is a phase, a small time period, which is called as interkinesis. Okay, I'll just write it here. Interkinesis. So in this phase, in some cells you will be able to see interkinesis. In some cells you will not be able to see the interkinesis. Okay, so basically it is like kind of a small duration. Maybe for some cells it will be like kind of resting for 1 hour and then it will start to divide. So that resting time is what we call it as interkinesis. Okay, so now let us try to write interkinesis. Some cells have a resting phase between meiosis 1 and meiosis 2. This phase is called as interkinesis. Nothing happens during this period. The cell just rests for a while. Okay, and one more thing, it is not seen in all cells. It is not seen in all the cells that undergo meiosis. Some cells will show this interkinesis, will not show interkinesis. Okay, so please copy this thing down. If you're done, let me know so that I can proceed further. Okay, so if I can proceed, I will move on further. Then we will go to meiosis 2. Okay, meiosis 2 is exactly like mitosis. Okay, meiosis 2 is exactly like mitosis, whatever we have studied. So what will happen in prophase 1? So karyokinesis 2. What will happen in prophase 2? So tell me, we already have studied mitosis, so which means you should know. What will happen to the nuclear membrane? The nuclear membrane disappears. Cell organelles what happens to the chromatin reticulum? Chromosomes la multiply agadu. A chromosomes will not multiply. That is what I am trying to tell. Before meiosis, nothing will happen. In the interface, no multiplication and all will not happen. Okay. Multiplication has happened already. So please, like, I think you are just confused. Please see to that. Let us say that this is a cell, germ cell, which is going to undergo meiosis. Then what will happen is this will undergo multiplication. After the multiplication only, what is happening? Meiosis 1 happens. And after meiosis 1, what is the phase that is there? That small phase is called as interkinesis. This interkinesis is there. And immediately after that, what will happen? Meiosis 2 will happen. Okay. So which means the thing will not happen. So which means like here, there is one cell. After multiplication, the cell is growing. And this is like, okay, during the multiplication, the cell is growing. And after that, it is divided into what? Two cells. And then this is like kind of going and dividing into what? Two cells. And then this is going and dividing into what? This is what is happening in meiosis 2. Okay, so which means the cell is here initially, whatever we have studied till now is meiosis 1, then it is undergoing this interkinesis, and then it is going to meiosis 2. Each of the cell will divide into two cells. Are we able to understand? So the chromatin fibers condense and spindle fibers arise from centriole. Okay, so that is prophase 2. Can we move on? Then metaphase 2, what will happen? The chromosomes are arranged in equatorial plane. The spindle fibers attached to the chromosome. You can write it simply. I mean, like, yeah, you can see the older thing and you can write it. But the whole thing is like, you just write some snapshot of what is happening. That is fine. Because since you are studying mitosis detailedly, it is the same thing which is going to repeat here. You can write it like a small hint kind of thing. You can write it. And then what happens in anaphase? The spindle fibers contract and pull the chromosomes to the opposite side. Yeah, anaphase promoting factor will cause the separation. Yes, you have to write whatever is being written. Please write it. Okay, so just a small hint. Anaphase, during anaphase, what will happen? The spindle fibers will contract and it will pull the chromosomes to the opposite side. And then the telophase 2. Which slide, ma? Who's asking? Martin, which slide? Now tell the number. Well, okay, I'm moving on. I just have 5 minutes. So, in telophase, what happens? The spindle fibers disappear. The nuclear membrane reappears. The chromosomes uncoils. A single cell will have two nucleus. Okay. So with that thing, telophase is done. And what will happen in cytokinesis 2? A single cell divides into two. At the end of cytokinesis, to four haploid daughter cells are obtained. It can be either through furrow formation or it can be through cell plate formation. In plants, it is cell plate. In animal cells, it is furrow. Okay, so this is what is bringing us to the end of meiosis completely. Okay, one important point that you have to write here is meiosis is a reduction division because dash cells are converted into dash cells. What is the first dash I will try to put? The first cell diploid is converted into haploid cells. The diploid cells are converted into haploid cells. Okay, dyads and haploids are not same. Listen, I will use the word diploid, haploid to indicate the number of chromosomes. Is to indicate the number of sets. It is one set. We call it as haploid. If it is two set, we call it as diploid. Okay, when I'm using dyad, tetrad, bivalent, they are all pertaining to only one pair. Okay, whenever I'm using the word dyad, tetrad, bivalent, all those things are connected with only one pair of homologous chromosomes along with their duplicates. Okay, whenever I'm talking about diploid or haploid, I'm talking about the entire set of chromosomes which are present in the organism. For example, I am a diploid organism because I have 46 chromosomes or 23 pairs in my body. Okay, so with this thing, meiosis is over, cell division chapter is also over. Okay, so please read this chapter. That is the homework. There is nothing specifically as homework. Please do read the concepts. Please do read the notes. Try to understand. Next class, we'll start the next chapter in biology. Okay. Thank you very much, children. Someone is asking for 11th slide. Yeah, you can see the 11th slide. Show all the slides from first. Okay, I will show it. Okay, I'll show all the slides. Please see. First slide. Second slide. Third slide. Fourth slide. Fifth slide. Sixth slide. 7th slide, 8th slide, 9th slide, 10th slide, 11th slide, 12th slide, 13th slide, 14th slide, 15th slide. Okay, so with this thing we are completing the chapter. Thank you very much.